Hello, my name is Angelina Sorensen. Hopefully you can see my screen and this is working like I think it will. But today I'm going to virtually do my transcriptome analysis walkthrough. Um, Hopefully Dr. RG has given some sort of primer on this, and you're relatively familiar kind of what I'm going to be kind of going through. Um, Typically, the last couple years, I've given this talk in person, so this will be my first time doing it digitally. So I figured the best way for me to do it would be to go through the slides like I normally would on one side of the screen, but then actually go through the live demo on the other side so that any, troubleshooting that has to happen, I can kind of do on the spot. 'Cause usually that's one of the benefits of doing it in person. So without further ado, I will just kind of go through these slides, which will be made available to you. And then I'll do it myself on this side of the screen, just so that you can kind of pause and follow along. Um, and yeah, so the way I would do it is I would just maybe have this video up and then have the slides up, and then just have another window pulled up. In advance, I have already um, also opened this Q value less than one data set. You'll have Q value less than one, and I believe Q value less than five. We'll talk more about what those titles mean in a second, but I would go ahead and open one or both of those. It doesn't really matter which one you choose. If one partner wants to do one partner wants to do another. I'm going to do less than one just because that's the one I happen to have open. But I already have that open. So I am going to go back to my screen here. Um, and yeah, so instead of doing a kind of general outline at the beginning, I'm going to kind of sprinkle that in as I go. I've included some useful terms here, which again will kind of come up as I go through the actual demonstration on the other side. So these are more just for your reference. I'll kind of touch on them when they come up. Um, so let's get into it. So the first thing you're going to do, like I said before, is open your RNA sequencing data sets. I only have this one open, but you can open one or the other. Um, so we're going to open that, copy all of the genes with a fold change greater than 4, or yeah, greater than 4. So for me, I'm going to open my original RNA-seq data. You can do up by gene or down by gene. Typically, I advise one partner to do up by gene, one partner to do down by gene, just so that you're covering both sets. So for me, if I do down by gene, I said here, copy all of the genes with fold change greater than 4. So see here, I copied only the genes, not the fold change. So I'm going to do is I'm going to copy all of these and I'm going to go all the way down until 4. Ignore any highlighted ones. Those are probably ones that I touched on for demonstration. So I'm going to do everything that's above 4, even though all of these are smaller. I'm going to do, so I have 545 genes selected for me. So I'm going to copy all of those. And then I'm going to go back to my windows here. And then we're going to go to either this link, which I think doesn't work anymore. Yes, so we're not going to use that link. But it's super easy. All you have to do is type in David database gene ontology, which I searched recently. And it will probably be the first one that pops up. It's the NIH logo. It looks like this. I'm going to click on it. And then, like I have in here, a shortcut to David Tools functional annotation. So they've updated the website a little bit in the last couple years since I've made this. So you should be able to go here and then functional annotation, or just when you open up the website, if you see all of these options here, this functional annotation will take you to the same place. So I'm going to go to functional annotation, and then going to go to the next slide. So we're going to paste the gene list you just copied. Going to pick official gene symbol, Homo sapiens, gene list for the list type, and then we're going to hit submit. So I'll go back to my window here and I will do that. I'm going to paste the genes that I copied, and it should show just like a list, just like it was when you copied it. We are not doing it from a file. For the identifier, I'm going to do official gene symbol because that's what we have up here. We have the gene symbols in our list. For species, we're going to do Homo sapiens because these are human cells that were used. And then we're going to do gene list, double check everything, and then we're going to hit submit. And the website looks a little different, but it shouldn't change anything too much. We gave you list one. And I switched mine to the classic version because it looks more like the way that I was taught, so I'm a little more comfortable with it. So if you don't want to do that, you don't have to, but I am going to keep it that way. So after you submit, you should see something that looks like this. You can rename the list if you would like. And then you should see the sub option that says gene ontology. So I'm going to make this a little bigger. Okay, so I am on this screen that says list report, and it's going to look a little different than this because it looks like they've updated the website a little bit to kind of look more modern. So to get to a similar looking screen, I'm going to go back up to the top here that says shortcut to David Tools, and I'm going to click on functional annotation again. And then we start to see more similar options to here. Again, the buttons are kind of different colors, they're in a different place. But we see functional annotation clustering, functional annotation chart, functional annotation table, and we see those same options here. So we're going to clear all so that all of these turn to 0. See if you check the defaults, it'll select some of these guys, but we want to clear all. And then we want to go to gene ontology right here. And we're going to go to go term BP, functional annotation clustering. Here we go. So here it has underscores on the slides, but here it looks like go term BP FAT, which means functional annotation clustering. We're going to click that. And then at the top here, I know here it says scroll down because that's how it was in the view before, but we're going to select go term BP, functional annotation clustering, and then we are going to click it right here. And then it's going to load for a second because it always does. And we're going to go to the next screen. And you'll see here, it shows you some clusters. You have different enrichment scores. And then we should see an option that says download file. I'm going to make this a little bit bigger. I'm going to switch to the classic version just so that it matches more kind of what we're expecting on the slides here. So you can do either or, but classic version is what I'm more comfortable with because it looks like what's on here. So I just switch it over to the classic version once I matched up with this spot here. And then now I can click download file and it's going to look like this mess of nonsense. And this for me is really hard to read. It's not all that useful. It doesn't really give me any information that I want. So I am going to copy all of it. So I'm just using keyboard shortcuts, but you can select everything and then just hit copy. And then you're going to go to a new Excel file. Or for me, I'm just going to go in the same key value less than 1, and I'm going to make a new tab so that I don't have to keep track of quite so many files. And I'm going to put it to the side so that I can have the slide next to me. And I'm going to right click, hit paste special, and then you should see an option for paste special again. And then you'll have 3 options. We're going to choose Unicode text. Okay. And then that will take all of this kind of chaotic nonsense and paste it into nice, shiny columns so that it's actually useful for us to look at. So go ahead and do that. And then we're going to go to the next slide. I put save this somewhere for if you made it into a new file. I'm just going to rename it on the same sheet. I'm going to call it David output. Yeah, that's all I'm going to call it. David output, I'll go 1 in case I do this again. So for gene ontology, this is one of the terms that I listed in the beginning. It loosely describes its function or a kind of broad header of what gene could be placed under. Like cell division could be a category, and any gene that makes a protein that has to exist in cell division or facilitate cell division in some way would fit under that broad category. And they can be part of a bunch of different categories. Categories can be very, very broad, like cell cycle, or they can get very, very narrow, like chromosome segregation and separation. So gene ontology terms can be very, very broad or very narrow. In the pasted version of the David output here, you'll have all of your genes and their different terms that they can be a part of. So here we have genes like nuclear division, which is pretty specific, and then cell division, which is a little more general, organelle organization. So there are tons of different kinds of Go terms. The numbers aren't that important. You won't have to know those. I would more pay attention to the word version of the ontology term, just so that you can kind of keep straight the different groups that these are organized in. So I said that there are many genes in each category, and those are based on the Go term or the gene ontology term. And the ones that we will be working with must have an FDR that's greater than 0.05. Post-production, Angelina here. In the recording, I just said FDR greater than 0.05, and I didn't remember that I had this typo in the presentation until I was watching it back to make sure that it was all right and cohesive before I sent it out for you all to see. So I say greater than 0.05, but then for the rest of the video, I proceed to say less than 0.05, which is what I actually meant. Sorry for the confusion. Wanted to put this in here so that you weren't like, why did she say greater than? And then she said less than for the rest of the video. Which one is it? It's less than. Ignore what she just said, but listen to what she says for the rest because it is less than. Sorry, and thank you. And an FDR is, again, one of those terms I've listed in the very beginning. You can think of it kind of like a p-value. If you remember back to biostats or any statistic class you've taken, you want a p-value to be a certain amount, usually 0.05, so that you have a specific amount of confidence that the data you're working with through the results you're reporting are statistically significant. An FDR is like a p-value adjusted for the amount of false positives that happen with genetics data. So you can think of it for all intents and purposes as a p-value. It just happens to be called an FDR when we're looking at kind of big genomic data sets like this. We're going to go to the next slide. And then this is totally optional. You don't have to do this. But for me, it was an easy way to visualize ones that had an FDR less than 0.05. So for me, I'm going to make this a little bigger. And again, the FDR column is this one that we care about. So it'll probably be the last column that gets pasted here. So if you want to do this so that you can visualize them a little bit better, all I'm going to do is I'm going to select this whole column. So I'm going to click this M up here for me. I'm going to go to conditional formatting, just like it says on the slide. I'm going to go to highlight cells rules and then less than. And then I'm going to choose less than or equal to. And I'm going to make it 0.05. And for me, I'm going to make it green because those are the ones that I want. Green with dark green text. And then you can choose any color you want. Doesn't matter. This is just for you. You won't have to turn this spreadsheet in. I'm going to hit OK. And then all of the ones that fit that criteria now will be green. So this one, for example, the FDR is 0.014. It's pretty low. This one is green, so I could choose any of these genes to validate or devalidate. And again, a little bit more on that in a second. But genes in this category, for example, this is much higher, so I would not want to choose genes in here. Now, if one of these genes is also in here, that is totally fine. If it's in a category that has an FDR within the criteria, it is totally within reason for you to pick that gene. So that's just kind of an easier way to visualize it. So I'm going to go to the next slide. Now, I didn't download this ahead of time so that if any troubleshooting needed to happen, I could do it live on the demo. So I'm going to go to Cytoscape. I have the link earlier in the slides. So I'm just going to hit download. We're going to do this together. I'm on a Mac, so I'm going to download the one for Mac. If you're on a Windows computer, obviously download the right one for you. And then it is going to load for a second. So while it's downloading, feel free to pause this, get it downloaded for you, make sure you know you're saving it, et cetera, et cetera. And once it downloads, I will start talking about it again. Okay, so my computer has downloaded Cytoscape. I clicked on it, clicked on Cytoscape installer here, and it brought up this error because it's from the internet. I've downloaded this before and it's not giving me any problems. I trust it. It's, from science, it shouldn't give you any problems. So I'm going to hit open and then it will think for a second. This is what popped up on my screen. It says, please wait. So I will give it a second to do its thing. Super fun. Cool. To verifying something. And then the Cytoscape setup wizard just popped up. So I'm going to go through the setup and hit next. I'm going to accept the agreement so that it lets me download it. I'm going to install it in my applications folder. If you're not familiar with the setup of your own computer, you can just have it download wherever the default is. That's where I'm leaving mine. You can create a desktop icon. I'm just gonna let it do what it wants. It is going to try to tell me some new updates, which I don't care about, so I'm gonna skip. And then it will finally start installing. So I will let it do its thing once again. There is usually some hiccups in the downloading of Cytoscape on different computers, so hopefully this is as complicated as it gets. If you are not able to get it to work, you can always borrow a lab computer from next door from the 208 lab, or one of the loaner laptops, or if one lab partner can get it to work. Maybe you guys just work together on one computer for now, if you can't get it to work. But hopefully this is all that would pop up. So it looks like it is finished. So I'm going to hit finish. And then it will once again think for a second and then hopefully open if we're lucky. So now it looks like it's at least in my computer. So I'm going to open it, see if it will open. It's always the hardest part for some reason, if it will open. We are still at this stage. And it looks like it is trying to open on my computer. This is why it's useful if you download it ahead of time and make sure you can open it. Um, but if not, sometimes it just takes a second and that's okay. It shouldn't take more than a few minutes. Um, so it is trying to open on my other screen and then I'll move it to the screen that is shared. So hopefully you're already having a lot of fun. So when it opens, it should look something like this. And then it will make your computer lag a little bit, which is fine. So come on. So here I say open Cytoscape apps bingo. So for me, I'm going to go to Cytoscape apps and then mine is already downloaded bingo because I've used this software before. But if you do not yet have bingo and you didn't do this in advance, you would just go to App Store, show App Store, and then it should be able to bring this up for you. And then if you looked up bingo, it will open another window, and then you should be able to just download it from the internet. Beautiful. So you just download it from here. I'm not going to download it again because I already have it. But once you download it and you know where you saved it, you could do the same thing. You just go to Apps, App Store, install app from file. And then wherever you had bingo saved, that's where you would upload it into the Cytoscape program. So I already have it, so I'm not going to do that. And I'm not going to let Cytoscape track me. So I'm going to go ahead and close that. So you're going to open bingo, and then I'm going to go to the next slide. So I'm going to go to apps and bingo, and then it's going to look like this, similar to the slide. I'm going to name mine Your Cute Jeans. That might be an outdated joke now because now I am old and I've graduated years past. But I'm going to name mine Your Cute Jeans. You can name yours whatever. We are going to get our Q value less than 1 or whatever spreadsheet you picked. I already have these still selected from when we copied them on the very first slide. So I'm just going to right click and copy them once more so that I can paste them in here. I'm going to go back here to this screen. Again, I know this is many windows. If you can do this at home where you have multiple screens, I definitely recommend doing that. Or if you are an iPad person and you can have an iPad with you and use Sidecar with your laptop, anything like that, I think is super helpful. Just because it is kind of a lot of windows to manage at this stage. But if not, it's all totally doable just in class during normal class time. It just does take some, some clicking around. So I've already copied my gene list, so I am just going to paste them here if it will let me. Copy. paste. There we go. So here I have my genes that are just from this spreadsheet. We're going to choose our significance level. It should already be 0.05 once you have the program loaded up. We're going to, I believe, not change any of the other fields, except we are going to change Saccharomyces cerevisiae because we are not yeast. We are going to choose Homo sapiens because that is what we are. If in grad school you decide to use S. cerevisiae, maybe you will choose yeast at this stage, but we will not be doing that. We're going to pick Homo sapiens because, again, these are human cells. So the yeast genes wouldn't make sense. They might have, gene equivalents, but they won't be named the same thing necessarily. And then I don't believe we will change anything else. I'm going to name mine your acute genes too, in case I already have one called your acute gene, since I've done this walkthrough many times. And then the last thing is save bingo data file in, and this is up to you, however you organize your computer. If you are not particularly organized, I would maybe go to your desktop, do a new folder, call this, Biochem 436L, bioinformatics stuff. Just so that you know where it is. You'll need this for your rationale paper when you get to that point. Hopefully, you will choose a gene and then you'll have to talk about this whole process and how you went through it on your own. So making a new folder could be useful to you. I would do it if it were me. So I just made a new folder on my desktop so that I can locate it easily, and I'm going to click open so that it populates in this little box here so that you know when you run it that it will save somewhere where you can keep track of it. And then I'm just going to click start bingo. Then it will load for a second. If you can see, my cursor is still spinning. So the output that has popped up for me looks like this. So I have this separate window that is the actual list. And then I also have on here this big graph of sorts with all of these little bubbles. I make this full screen and I zoom in on it. If it will let me. Yeah. If I zoom in on it, I can see that all of these little bubbles actually have Go terms in them, the little categories that we talked about earlier. So the good thing about this is that you can move all of these guys around. If you see here, some of the names are pretty long and they get kind of jumbled in here. So if you want to see what it actually says, if it's kind of in this mess of things, you can drag them around. The way they're organized doesn't really matter, like where they are spatially with one another. What matters more is the connections. So that's why you're able to move them. Like if I move this guy way over here, these two are still the ones that feed into it. That relationship doesn't change, just if you move it, farther away so that you can read what it says. So you will need a picture of this for your rationale paper. So once you kind of zoom in on a subject or one of these categories that is meaningful to you or that you're interested in, if you want to kind of move these around a little more so that they look a little better, that it's easier to look at. Like if you turn something in like this where you can't really see any of the terms, that probably will not earn you favor for your grade. But if you are looking at, this section, for example, these are all spaced out nice. It's not cluttered. It shows, kind of the progression of what you're looking at. So that would be a lot better. But feel free to kind of manipulate these and move them how you want. The size of the bubble correlates to kind of how many genes are in that particular category. So you can see biological process is kind of the big umbrella term for all of them, and that's the biggest bubble, rightly so. Response to abiotic stimulus is a lot smaller. Response to X-ray specifically is even smaller. So this, if you click on the bubble, it will show you how many genes are actually included in that. um, down here. And it will show you the adjusted p-value. Um, and yeah, so you'll have this graph. I would take a screenshot of it, save it somewhere so that you can come back to it. And then you also have this, which is the actual output, and it shows you the Go description. What I would do to take a better look at this is I would copy all of this. I think you can just use the keyboard shortcuts. So for me, I'm hitting Command A on a Mac. I think it's Control A on a Windows computer. And I'm just going to copy them. And then I'm going to go in our same spreadsheet, and I'm going to rename it as bingo output. I'm going to move it after the David output so I remember the order of them. And I am just going to paste it and see if it retains its formatting. And it did. Cool. So I'm going to insert a row up here so I can add these up top. So that I can add these are our genes. These frequency, cluster frequency, these don't matter quite as much. We want our Go description. These we can hide just for simplicity's sake. So there are a couple different ways to go about picking a gene. One of the most common questions I tend to get at this stage is what do you guys mean by validate or devalidate a gene? Like what does that actually mean? So let me add a new slide in here so that I can give you an example. New slide. So what does validate or devalidate actually mean? How can you validate or devalidate a gene? So what we mean by that is the PCR findings um, that you're going to be generating this semester in the lab. Are you expecting that to match this data? If we go here, do you think it'll match here? Or do you think your PCR data will look different? Like, am I validating that GNB4 is downregulated? Do I think that that is true? Or do I think, that doesn't make much sense? I'm going to devalidate this finding. I think when I do PCR, that it will be the opposite direction, that this gene will be upregulated. That would be devalidating. That's an example of devalidating. So if hypothetically, I tell you that there is a gene called there's a cancer gene, and that's what it's called. And in the RNA-seq data, you see that the cancer gene goes up in response to the caffeine and pomegranate juice treatment. Does this make sense? You don't think too hard about what the cancer gene actually does, just you know that it causes cancer, that it is a pro-cancer gene. And when you administer the anti-cancer treatment, right? You know the basics of kind of how it works. When you administer the anti-cancer treatment, the cancer gene in the RNA sequencing data goes up. That might be something that you want to devalidate. You would say, oh no, I think the treatment is an anti-cancer treatment. It, inhibits all of these things. You know, it makes migration happen less. It makes the cells adhere better to each other so that they can metastasize. I think it would make more sense for the cancer gene to go down. You would think that there would be less of it. if you're giving a cancer killing treatment to the cells, right? Maybe, maybe not. That's up to you if that makes sense to you. Um, so that's kind of what we mean by validate or devalidate. Some of them are pretty straightforward, like the cancer gene, you would think, you'd think it would go down. There are some that, maybe there's a post-translational modification that you don't know about. Maybe RNA goes up, but, some other modification happens down the line. And that's why you'd want to devalidate it because you would say, oh no, I think it's actually going to look like this. Because PCR and RNA sequencing are, they can a lot of the times go in the same direction, but they might not, depend on, what you're looking at. So that's one of the questions. Another question is kind of, how should I pick a gene? And I think I have a little bit about that in here. Um, so here I just talked again about those bubbles, um, the bubbles that are on this screen. So again, everyone's going to look a little bit different. I put a little bit of information about what they mean. Like I said, the size of the nodes is dependent on the number of genes associated with that particular description. And then another thing that I did not mention is that the darker the bubbles are, the more statistically significant the genes in that category are. So if we go back to the cytoscape window, these ones that are orange as opposed to yellow have more statistically significant genes that were differentially regulated, which just means they were, the expression was different as opposed to the before stage, which in our case was before the treatment. So that is something to note. And then if I go to the next one, go terms may be different, like I said. These kind of change as more genes are, identified, and more jobs of genes are uncovered. There are some genes that, we know one of their functions, but we don't know everything that they do. And so things are always being updated. So what it looks like in this slideshow probably isn't exactly what this looks like, and it's not going to be exactly what it looks like when you do it, such is the nature of science, and that's okay. So don't worry if yours looks different than your partner's, if it looks different than mine, as long as you're seeing the same kinds of windows and the same kinds of steps, chances are you're on the right track. Final remarks, pick a gene you think is cool. It'll make it a lot more fun for you in the end. If you pick P53, chances are you know a lot about P53 already. You've probably studied it in maybe 208 in cancer bio. If you took it, I chose a gene that I had never heard of and I didn't know anything about. And I got to become an expert about it with my lab partner, and it was a lot more fun. So if you're really interested in neuro, if you're really interested in metabolism, chances are there's a gene in here for you. There was a group a couple years ago who was more interested in the neuro side, and they picked a dopamine transporter or serotonin transporter or something like that. And that isn't something you would think to associate with cancer, but it was, it was upregulated or downregulated in this data set. So it was something that they were free to pursue. So think about, talk with your partner about what you think is cool, what you'd want to work on, because that will make it more fun to do as opposed to, ugh, I have to do this for class. This is stupid. You know what I mean? Um, Save everything. You will need to talk about all of this for your rationale paper. I'm not there to talk about it with you, but Dr. RG will talk more about the expectations of the paper. Hopefully this is a good resource to you to be able to reference back to. I've even included, me talking on the recording so that hopefully that is helpful for accessibility purposes. It's easier to, follow along. If anything doesn't make sense, definitely ask Doctors RG and Basafi. They'll help to the best of their ability. If not, if there's a question they can't answer, I'm still around. You can always shoot me an email and ask me. I will add it here, Angelina's contact info. You can always shoot me, a message on LinkedIn and ask me. and I'm happy to help. Or you can reach me at my USC email. Oop, misspelled my own name at ufc.edu. Um, and if you had some technical difficulty and you weren't able to go through it, I'm happy to hop on a call at some point. If we can find a time that works out. If not, you can always send me a screenshot and I'll do my best to help. Um, I really enjoy this project a lot. I care a lot about it. So if, if anything comes up, just know that I'm still here, I'm still around, as a previous student, as an alum. Um, and yeah, that is pretty much all that I have. Um, so yeah, hopefully this was helpful. Um, do tell feedback to Dr. RG if it's not helpful. This is the best way I thought to do it so that you can actually see what I'm doing while I went through the slides. So hopefully it was helpful. Feel free to email me feedback if you think I should make a change. Um, this only comes around once a year, so maybe I can do another recording before next year. Who knows? But yeah, that is all I have. I'm going to stop sharing. Um, yeah, I hope that this was helpful. Um, please let me know if it was or was not. I'm around. And have fun. You've made it to almost the end of your degree, so that is something to be celebrated. Congrats. I'm rooting for you, and I hope that you have fun in this class like I did. Peace.